Energy transformations in oxidative phosphorylation. Most cellular energy in eukaryotic cells is generated in the mitochondria through the process of oxidative phosphorylation. This process consists of a series of energy transformations that result in ATP synthesis. In mitochondria, ATP synthesis begins when redox energy from the electron transport system is used to move protons across the inner mitochondrial membrane, creating a proton gradient. This proton gradient is a source of electrochemical energy, which is converted into chemical energy as protons move through the ATP synthase complex, generating ATP. Let's take a closer look at each of these energy transformations. The process of oxidative phosphorylation requires the electron transport system, consisting of 4 protein complexes and an ATP synthase complex embedded within the inner mitochondrial membrane, as well as associated electron carriers. The ongoing synthesis of ATP in the mitochondrial matrix also requires 2 transporter proteins that provide inorganic phosphate and ADP for the reaction, while at the same time exporting the newly synthesized ATP. The electron transport system captures energy from a series of sequential redox reactions. Oxidation of NADH and FADH2 releases electrons. We will follow electrons from NADH, which enter at complex 1. These reactions are coupled with the movement of protons across the membrane. The electrons flow through the electron transport system. And reduce oxygen to form water. The redox energy released by these reactions is used to translocate protons from the mitochondrial matrix to the intermembrane space, creating a proton gradient. Protons move back into the mitochondrial matrix through the ATP synthase complex, generating ATP. This movement of protons across the membrane down their concentration gradient to generate ATP is called chemiosmosis. Directional proton translocation by the electron transport system increases the proton concentration on one side of the membrane, creating a proton gradient. This proton gradient is a source of electrochemical energy due to both the chemical gradient, pH differential, and electrical gradient, charge differential. Chemiosmosis takes advantage of this electrochemical gradient, which causes protons to flow back into the mitochondrial matrix through the ATP synthase complex, providing energy to drive ATP synthesis. The electron transport system can then recycle these protons, completing the proton circuit. In plant chloroplasts, chemiosmosis is also used to synthesize ATP. Chloroplasts use sunlight energy rather than redox energy to establish a proton gradient across the thylakoid membrane. The proton circuit behaves like an electrical circuit, as potential energy is converted to work as a result of movement of charge. Proton circuit uncouplers disrupt proton flow by allowing proton leakage back across the membrane without the usual production of ATP. This converts redox energy to heat instead of ATP synthesis. Certain compounds, such as oligomycin, can inhibit the ATP synthase complex, blocking the proton circuit so that oxidative phosphorylation cannot occur, eventually leading to cell death. We can see that each part of these energy transformation processes is vital to sustain life.